Glycolysis, which is also known as Embden-Meyerhof pathway. Glycolysis is the series of enzymatic reactions where there is oxidation of glucose into pyruvate in an aerobic condition and glucose into lactate in an anaerobic reaction for the generation of adenosine triphosphate ATP, which is a form of energy. Glycolysis occurs in cytosol of every cells of the body, and for the erythrocyte it is the major source of energy. However, anaerobic glycolysis is important to provide ATP molecules to the skeletal muscle in hypoxia and allow the tissue to survive anoxic episode, that is anaerobic condition, and cardiac muscle adapted for the aerobic performance poorly survive under ischemic condition. Therefore, cardiac muscle undergo aerobic glycolysis. Moving on to glycolysis, in initial step there is phosphorylation of glucose molecule by the enzyme hexokinase to glucose 6-phosphate by the utilization of energy, that is ATP molecule. So once the glucose molecule is transported inside the cell, it is immediately acted upon by hexokinase to convert into glucose 6-phosphate, which means there is the prevention of leakage of glucose from the cells. Glucose 6-phosphate is then converted into fructose 6-phosphate by the enzyme phosphohexoisomerase. Here the reaction is reversible reaction, but fructose 6-phosphate is immediately utilized in the next step. Therefore, the reaction is directed in forward direction. Fructose 6-phosphate is then converted into fructose 1,6-bisphosphate by utilization of ATP in the presence of enzyme phosphofructokinase. So the steps up to here in glycolysis, these steps are also known as energy investment phase because there is utilization of 2 ATP molecules. Up to now there is no production of ATP molecule, rather utilization. Hence it is also known as energy investment phase. In next step, the hexose structure of fructose is splitted into triose sugar, that is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate by the enzyme fructose bisphosphate aldolase. This stage is known as splitting phase because the hexose sugar, that is fructose, is splitted into 2 triose sugar, that is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So one glucose molecule gives us 2 triose sugar which undergo the energy generation phase to give 2 NADH and 4 ATP molecules individually. So these 2 triose sugar that are in equilibrium by the action of phosphotriose isomerase or triose phosphate isomerase. However, glyceraldehyde 3-phosphate is continuously utilized in the next step. Hence, dihydroxyacetone is continuously converted to glyceraldehyde 3-phosphate to maintain the equilibrium. The triose sugar is then acted upon by glyceraldehyde 3-phosphate dehydrogenase for the production of 1,3-bisphosphoglycerate. The reaction is dehydrogenation reaction and there is production of 2 NADH molecule in this step along with addition of inorganic phosphate. In the next step, there is enzymatic transfer of phosphate from 1,3-bisphosphoglycerate to ADP to form ATP in the presence of enzyme phosphoglycerate kinase and the product 3-phosphoglycerate. Now this 3-phosphoglycerate in the presence of phosphoglyceromutase is converted into 2-phosphoglycerate. The next reaction is elimination reaction where H2O is removed in the presence of enzyme enolase forming phosphoenol pyruvate. Now phosphoenol pyruvate is acted upon by pyruvate kinase to produce pyruvate along with the production of ATP. So in the final step, usually in the muscle, pyruvate is acted upon by lactate dehydrogenase where there is conversion of NADH to NAD, finally forming lactate. Now we'll move on to energy yield in glycolysis. For this, we will first remember the 2 ATP molecules that were usually invested during the energy investment phase. We'll start with aerobic pathway. In aerobic pathway, there is production of 2 NADH molecule at glyceraldehyde 3-phosphate dehydrogenase. We need to remember 1 NADH molecule is equals to 3 ATP. So the total production is 6 ATP. In phosphoglycerate kinase, there is production of 2 ATP and in pyruvate kinase, there is production of again 2 ATP molecule. So the total will be 10 ATP molecule up to here. but we need to subtract 2 ATP molecule that was initially utilized in energy investment phase. Hence the final product is 8 ATP molecule. Now moving towards anaerobic pathway. Here we will not add 2-NADH at the step of glyceraldehyde 3-phosphate dehydrogenase, because these 2-NADH will be later utilized in the step where there is formation of lactate from pyruvate. Basically, there will be 2 ATP production at phosphoglycerate kinase and 2 ATP at pyruvate kinase, giving up of total of 4 ATP molecule. Again, we need to subtract 2 ATP molecule that was initially utilized in energy investment phase. So final product is 2 ATP molecule in anaerobic glycolysis. So that's it all about the glycolysis, its energy yield. I hope the video will help you.